Hello madam, my name is Darshini Satyamurthy from DBT3A. My metric number is 23 DBT20F1025. Today I am going to do a lab simulation based on Agarose Gel Simulation. Agarose Gel electrophoresis is a method of a gel used in molecular biology to separate a mixed population of macromolecules such as DNA or protein in a matrix of agarose, one of the two main components of agar. Moreover, agarose gel electrophoresis is commonly used to separate DNA fragments following a restriction digest or PCR amplification. Now start the simulation. Before I start the lab simulation, I will choose lab level 3. This, this is the context of this lab simulation. In this simulation, I will conduct gel electrophoresis to separate DNA solution of dye. I need to know before this lab start. Gel electrophoresis is used to separate biomolecules such as DNA and RNA in an agarose gel composed of tiny pores according to their size and electrical charge. An electrical current applied to the gel migrates the charged biomolecules toward the oppositely charged electrode. Since the DNA fragments have a similar negative charge, they all move toward the positive electrode. Smaller molecules will move through the pore in the gel faster than larger molecules. Lastly, loading dye is often used to visualize how fast a gel is running and add weight to your sample, allowing it to sink to the bottom of the well. These are the materials we needed in this experiment. This is the prediction of this lab simulation. In this simulation, I will be using a gel electrophoresis to separate three different dyes. These dyes differ in size, with the yellow dye being the smallest, the blue dye being larger, and then the purple dye being the largest. If these dyes were all equal in negative charge, I for the predict the dye will move in their line during the electrophoresis. Now I am going to drag the different dye to a position. Now start the simulation. Select the Agarose gel. Place the gel into the electrophoresis box. Orientate the gel toward the negative electrode. Pour the flask 1 sodium borate buffer into the gel box. Now close the electrophoresis box. Going to next. Open the microcentrifuge. Move all the tubes into the microcentrifuge. Now balance the microcentrifuge. Centrifuge look balanced and ready. Briefly the spin the tubes for 30 seconds. Put the tube back on the rack. Make a note in your notebook to show which solution is piped into each line. S1 to line 1, S2 to line 2, S3 to line 3. Now safe note, select the P20 micropipette and set it 10 microliter. Safe volume. Open the tip box. Attach a tip. Place the micropipette in the S1 solution tube. Move to line 1. Pipette solution 2 into line 2 following the same process as before. Now cover the electrophoresis box. Connect the red and black cable to the power supply and the lid of the gel electrophoresis box. Now turn on the power supply. Set the voltage to 130. Set a timer to 10 minutes. Start the timer. Wait until the time has elapsed. Now turn off the power switch. Unplug all the electrodes. This is the result. Explain why the band end up in those relative positions. In this what I predict will happen. The feedback result. What part of the procedure I think had the greatest impact on the result I obtained? I think at the pipetting solution in the lens part might be a reason for the impact on the result. It should it it should stop at the first stop. If it stop at the second stop, it might affect the results. This is the question below. The first question is, your lab partner put the gel into the electrophoresis box as shown below with the well oriented toward the positive electrode. So you run the same experiment as conduct in the simulation with the S1, S2 and S3 samples. How will this impact your result? So I will choose is mm, the dye will move toward the positive end and quickly run off the gel surface, resulting in an empty gel at the end of your experiment. The second question is, which is the best explanation for why the S1, S2 and S3 samples show different numbers of bands? The S2 solution was a mixture of dye molecules of different size and each band is the collection of molecules of a particular size. The third one is, what will happen if you increase the length of time a gel is run during a gel electrophoresis experiment? The molecules in the sample will run further through the gel. Why might you run the gel electrophoresis with an agarose gel like the one pictured below, where the well are in the center of the gel? If you want to separate molecules of unknown charges, the well in the middle gives molecules option to travel in both directions. The last question is, if you use gel electrophoresis to separate samples of DNA and get bands in different positions on the gel, what do you know about the molecules in those bands? The molecules are different sizes and some fragments have longer DNA sequence than others. The last one is feedback question. How confident do you feel about using the technique you learn? I I am feel about using the technique I learned is very confident. This is the summary below of this lab simulation. It is always good to summarize your methodology and observation after you have completed an experiment. Please view a recap of the simulation and takeaway messages regarding the running of a gel below. In this simulation, I learned the correct way to load and run gel electrophoresis in a lab. This is a technique that is frequently used in molecular biology lab. The simulation explores how to set up an electrophoresis box, use a micropipette to transfer liquid to the gel in the electrophoresis box, run a gel using electrophoresis to separate molecular fragments based on size and charge, interpret the bending pattern you obtain after gel electrophoresis. After that, this is the takeaway messages. Make sure your gel is always orientated with the well closest to the negative electrode if you running DNA or other negatively charged molecules. If the well are closest to the positive electrode, your samples will quickly run off the small piece of the gel above the well and be lost to the buffer. The second one is make sure there are no air bubbles in your tip before you load the samples into the well. If there are air bubbles, they will displace your sample and may lead to contamination of other wells. The last one is the risk of disrupting your sample and potential contamination is the reason you only proceed to the first stop when loading samples into a gel. Proceeding to second stop force air out of the micropipette tip which could displace your samples from the well. Now end the simulation. That's all from me. Thank you, madam.